First up, we have Dr. Zhiqiang on, Professor and Director of the Texas Therapeutics Institute at UT Health. He was a part of the original MIRA that was funded years ago and has been a part of our IDDD steering committee. One of those that graciously hosted the field trip to carry people through his lab last year and show the exciting equipment and capabilities that they have over there. He joined UT Health in 2009 after 15 years in the pharmaceutical industry. He's appointed Professor of Molecular Medicine, Robert A. Welch Distinguished University Chair in Chemistry, and Director of, as I said, the Texas Therapeutics Institute. He previously served as the Chief Scientific Officer at Epitomics and was Director of the Biologics Research at Merck. He started his biotech pharmaceutical career at Millennium Pharmaceuticals, where he worked in the field of biocombinatorial natural products, drug discovery, using microbial molecular genetics and engineering approaches.
Good afternoon, everybody. So today we will talk about antibody drug discovery. So I will provide an overview of the landscape of antibody as a drug modality, and then talk about the capabilities in my lab, and show some examples how we go about antibody drug discovery and development, and then ask you to dive deep into antibodies targeting multi-span complex memory proteins such as transporters, channels, and GPCRs. And look at, for those of us interested in drug discovery, drugs came with different modalities, starting from small molecules to peptides, proteins, and antibodies. In this case, I use the example of virus-like particle for an HPV vaccine. And more recently, we have drugs, gene-based therapy, cell therapies, and even nucleic acid-based therapeutics. So all these drug modalities are in clinical use and, of course, in clinical studies. So for people working in drug discovery, there's no better time to get into this field because it's just these are gene therapy, cell therapy, and RNA-based therapy just became drugs last couple of years. So my lab working on antibody as a drug modality. And when you talk about antibodies, so let's first review. So antibody is a natural molecule, and actually is a natural drug, which immune molecule that against infection. And so the antibody is a heterotetramer, has two identical heavy chain and two identical light chain. The variable region of the molecule engage the target, so you interact with the antigen. So for example, similar to a small molecule that bind to an enzyme, for example. So this can inhibit signaling, for example. But more than that is the antibody has this Fc region, which can mediate immune effector function, such as ADCC. That stands for antibody-dependent cellular cytotoxicity. And antibodies are heavily glycosylated, is actually very tightly regulated. So glycosylation impact the antibody immune function at half-life. And antibody, the Fc region can also engage the C1q immune cells that mediate complement-mediated toxicity. This is very important for the immune system to clear bacterial infection. And antibodies can also interact with Fc gamma receptors, which also mediate immune effector functions such as ADCC. Antibodies, in terms of IgG, came with different isotypes, IgG 1, 2, 3, 4. For drug development purposes, we often use IgG 1, 2, and 4 as a scaffold, which I'll get into this a bit more later on. Last but not least, the antibody Fc region can engage the neonatal receptor or the variable receptor. That's actually mostly contribute to the long half-life for antibodies. So human antibody, IgG antibody, has an average half-life of 21 days. So when you talk about antibody drug modality, you talk about antibody as a monoclonal antibody. And antibodies can also conjugate with toxins, such as chemo drugs in the form of antibody drug conjugates. And antibodies can also conjugate with antibiotics that for treatment of a bacterial infection. And antibodies are also conjugated with radioisotopes for targeted radiotherapy. And antibodies, by definition, they are very specific towards antigen, but we can engineer antibodies with multi or bispecific activities. So one antibody molecule can target multiple antigens at the same time. And last but not least is chimeric antigen receptor T cell therapy. The key component actually is also antibody. So in my mind, that T cell therapy is also belong to the antibody drug modalities. So antibodies, as I said, came from different isotypes. IgG1, 2, 4 are commonly used. And we can also engineer antibody efficacy with enhanced half-life or reduced half-life in some cases. And we also can engineer antibody efficacy with enhanced immune engagement or reduced immune engagement, depending on the purpose of your drug design. And we can also use antibody fragments, nanobodies. Antibodies came from camel or from llama, different species. There's only heavy chain, no light chain, so it's called nanobodies. I already talked about antibody conjugated with drugs, antibody drug conjugates. And the antibody also can conjugate with proteins. For example, recently there's an approval of antibody conjugated with cytokines, like IL-12, for treatment of cancer and immune diseases. And antibody as a drug source can come from animal sources, can come from synthetic antibody design. If antibodies came from animal source, need to be humanized to avoid immunogenicity in the clinic. And if the antibody are good enough in terms of affinity, but not good enough as drug molecule, we can enhance the affinity through called in vitro affinity maturation, similar to a medicinal chemistry for small molecule drugs, where you have a drug that's good but not good enough as drug molecule. And laborious immunization can generate antibodies. Animals can include mice, rabbits, non-human primates, even antibodies directly from human sources. And the nice thing about antibodies that is because the efficacy region of the molecule and also the variable region of the molecule, it can have different modes of action. For example, they can be agonists, they can be antagonists, they can engage the immune effector functions, and they can also engage T cells, they can do receptor internalization and antigen depletion, to name a few. Oftentimes, antibody molecule could have a combination of more than one modes of action, which I'll give you one example later on. So today, this is a slide borrowed from Dr. Bill Straw, who was an antibody expert. So he, in December, he summarized this. So basically, today there are 97, close to 100 antibody-based drugs in clinical use today. And there's a similar number of antibody drugs in late-stage clinical studies, phase 2, phase 3. And more importantly, you see there's more than 700 antibodies in phase 1 clinical studies, phase 1 to 2. And there are 2 CAR T cell therapies approved for clinical use today. You can tell there's more in the clinic. So this is a very active area of research, T cell therapy. But as I said, for T cell therapy, the key technology actually is the antibody you use. Of these, there's only about 100 validated target. So this just to show you that we still have a limited of targets to work on. And the success of antibody as a drug modality is reflected is a marketplace. So in 2018, the top 10 selling drugs, 7 of these top 10 are antibodies. So this is hard to believe. Antibody drug modality only less than 30 years, and already 70% of the best selling drugs are antibodies. The first one is Humira, targeting TNF-alpha for autoimmune diseases like rheumatoid arthritis, and Keytruda antibody targeting PD-1 for immunotherapy, and you have antibody Herceptin targeting HER2 for HER2 positive breast cancer, Avastin targeting VEGF for angiogenesis therapy for colon and other solid tumor, rituximab targeting CD20 for B-cell lymphoma, and Opdivo, another antibody from BMS targeting PD-1. And number 10 is actually antibody from Johnson targeting interleukin-12 and interleukin-23 for autoimmune diseases such as psoriasis. And also I want to point out to you that these targets are very simple soluble proteins, simple memory proteins like tyrosine kinase receptors. This became relevant because if we want to expand the drug targets, we need access to GPCRs, transporters, and ion channels. That's where Russ will come here today to talk about these difficult targets. And so today, the antibody cancer immunotherapy research is one of the hottest areas in biomedical research, as such that in 2018, the Nobel Prize for Medicine given to scientists, Dr. Harjo, who discovered PD-1, and Dr. Alicia here in Houston worked on CTO4, both immunotherapies. And surprisingly, in 2018, the Nobel Prize in Chemistry given to 3 scientists who work on protein engineering, and 2 of them actually, Dr. Smith from University of Missouri in 1985 published a single-page science paper displayed a peptide phage, that peptide still has binding activities against this target. And 5 years later, in 1990, Dr. Greg Vitter at the MRC lab in Cambridge, UK, displayed a single-chain fragment antibody on the phage that show body activity. And of course, Dr. Arnold is working on gene shuffling for protein engineering, which is used in antibody fetal maturation commonly. So, with all this good news and rapid development of antibody drug discovery, but there are major challenges facing the antibody drug development today. The number one challenge in my mind is the lack of novel antibody drug targets. As I showed you earlier, today there's only 100 novel targets be targeted. Because of the lack of validated drug targets, the pharmaceutical, the biotech industry are repeatedly looking for drugs targeting the same targets. Those targets are, you might give drugs that's maybe improved, but to me it's kind of incremental. So we should discover novel targets and truly to fulfill the medical needs of many diseases which are lack of drugs. For example, today there are of the 37 antibodies for oncology indication. 6 are targeting CD20. So the drugs target the same target, the same indication. There's some improvement, for example, you improved immune effector function, maybe higher affinity, maybe better potency, but it's just incremental. Not there's 6 antibodies targeting EGFR HER2 axis, and 6 antibodies targeting PD-1, PD-L1, the pathway. And it's a very surprising to me, reviewing this literature. Last year, 2018, 2019, there are 156 new antibody drugs into clinical phase 1 trial in the United States. This is the clinical.gov database. 15 of them are targeting HER2. We already have a trastuzumab, which already, of course, this is used for bispecific, also antibody-drug conjugation, but still the recycling of all targets is alarming to me. And also you have CD3, 16 of these, PD-1, PD-L1, 21 new phase 1 entry in United States law targeting this same target. We really have very good drugs you can use today. So this discovery of novel targets rely solely academic labs. So that's where drug discovery really is not a job for the companies, but it's academic labs actually discover drug targets. And next is lack of biomarkers. For example, the target IGF-1, HER3, those are, today there's at least dozens of clinical candidates in clinical trials. If you look at these clinical studies, statistically they are not significant, they are not effective, they are not efficacious. But some of the patients has a complete response. So we do not know why these patients respond, so that you need a biomarker. And if we sit here for PD-1, PD-L1 for the best indication is for skin cancer, melanoma, there's only about 20 to 40% of people respond. And the majority of patients are not, so we do not know why. So biomarker discovery is actually even harder than discover or validate the drug target. So this is when you talk about the basic research, to me, biomarker discovery is the most important area of research in drug development. And similar to small molecule drugs, cancer or tumor can generate resistance to antibody therapies. So we need to find a way to overcome drug resistance. Again, this is a very fundamental research. And these drugs has very bad clinical side effects. For example, the PD-1 antibody have, because the activation of T cell, so overactivation of T cell give you a lot of very bad side effects. For example, these antibodies, T cells can also kill beta cells, can induce diabetes, which is a very big problem. And so we need to find a way to reduce side effects, or maybe new therapies to mitigate the clinical side effects. And in terms of technology, antibodies by design, they cannot get inside the cell. So all the intracellular targets are off-limited by antibody as a drug modality. And antibodies cannot readily cross the brain-blood barrier or eye-blood barrier. So the central nerve drug targets like Alzheimer's and Parkinson's are off-limited by antibodies. So there's both industry and academic labs active programs to devise new technologies to get antibodies delivered inside the cell, inside the brain, and access to more targets. And I told you already, antibody engineering is a big part of the antibody drug discovery. So you can engineer antibodies to penetrate tissue, deep into the tumor tissue, cross BBB, get into the brain. And antibodies with enhanced immune effector function like ADCC, CDC, or antibodies with reduced immune effector function in the case where you do not want to activate the immune system too much. Antibodies already have a very long half-life, but you can engineer antibodies with longer half-life. For example, there's antibody targeting RAG ligand for called denosumab for osteoporosis. The half-life is 2 months. So you only need to give the drug every 6 months. So once every 6 months. And the antibodies can be also engineered as enzymes with the catalytic activities. And antibodies can be conjugated with drugs, with antibiotics, with radioisotopes as a carrier. And of course, it's the whole field of antibody immunotherapy, including T-cell therapy. Engineering is not trivial, but everybody can do this. But the most important thing is actually we need novel targets, we need biomarkers, we need better designed clinical trials. This came from basic research. So I was always in my lab say we are antibody engineer lab, but this is not important. This is important, but it's not the key. The key actually is the biology. And recognizing this, so in 2015, so we were awarded with a separate co-grant to support the monoclonal antibody drug discovery in Texas. And last year, we got another separate call, this time focusing on advanced antibody modalities. This includes biospecific antibodies, antibody drug conjugation, and so actually this, I think my lab is the only lab has two separate cores running in parallel. But they really focus on one thing, that's the antibody as a drug modality. So your summary is basically in the middle we have the monoclonal antibody technology, which is antibody from different sources, from B cells, from different animals, from libraries. And the antibodies can be humanized if from animal source. If antibody is good but not good enough, we can do in vitro affinity maturation to make the antibody better. The new core will focus on antibody drug conjugation and biospecific antibodies and antibodies for chimeric antigen receptor T cell therapies. So currently we have more than 20 drug discovery projects in collaboration with Texas-based institutions and also industry and labs outside of Texas. In terms of drug discovery, we focus on cancer because it's separate, and also antibodies. More than half of the antibody drugs today are targeting cancer. So we have a program targeting cancer-induced cachexia, muscle-based, lung, ovarian, colon, breast, acute myeloid leukemia, GBM, and pancreatic cancer. And then we also have a program targeting metabolities such as diabetes or obesity, antibody program targeting spinal cord injury, and drug-induced acute liver toxicity, and antiviral antibodies, and last but not least, Alzheimer's disease. So what I list here, they are all supported by multiple grants from NIH, from separate from DOD. So now I'm going to shift gears to walk you through one example how we go about antibody drug discovery. So this is a program in collaboration with Alec Zhang's lab in UT Southwestern. So Alec is a cancer biologist working on a group of receptors called the LILB family. They are actually, so this receptors short for leukocyte Ig-like receptor family. So number B4 is one of the receptors. So these receptors, intracellular, they have R-team domain, so the hallmark of immune checkpoint inhibitory receptors. Extracellular, they have Ig-like domain, and so very similar to PD-1. So this is a receptor has no mouse homolog, so that's why nobody studied it. So Alec's lab is actually the only lab actually studied this receptor. And so we worked on this for more than 7 years to identify, so we know that the extracellular Ig domain that can bind to linkage. When we study this program, is an awful receptor with one of those linkage. And intracellularly has the R-team domain that can recruit phosphatases such as SHAP1, 2, and SHAP that can activate the cascade for immune inhibition. And then these receptors are expressed on immune cells. They have immune checkpoint proteins similar to PD-1. They inhibit the myeloid and lymph node immune activation. But different from PD-1 or CTL4, these receptors can express leukemia cells or cancer cells. They can support cancer cell development, so they become oncogenes. So they're both oncogenes and immune checkpoint inhibitory receptors, depending on where they express. And they are this particular B4 receptor B4 is a marker for monocytic AML, which is called for majority of the pediatric AML, which has no cure. And we also identified one of the ligand for this receptor, ApoE. So this receptor knockout has no significant role in development, suggesting it could be a safe target for drug development. So all this work was published in Nature last year. So we established the biology, basically validated this is a novel drug target. So in the meantime, we started doing antibody development. So in this case, we immunized the extracellular domain of the receptor, B4 receptor, we immunized rabbit. And from here, we just collect some PBMC, the blood samples from the rabbit. You do have to sacrifice the animal. And then you isolate the memory B cells from these immunized animals and put it into single memory B cells by cloning of single cell antibody encoding genes. Recombinantly, you can express the 293 cells. And because it is antibody from rabbit, we have to humanize it. And so we show it has a binding affinity, the single-digit nanomolar, show a very potent antibody. And we crystallize the receptor protein and the antibody show where it binds, to study the structure activity relationship, very similar to small molecule drug discovery. And I told you this receptor is an immune checkpoint inhibitor receptor, so they can inhibit the T cell activation. So in this experiment, so if you mix cancer cell THP1, this is a leukemia cell line, and you mix with T cells, human T cells, and you add a control antibody, the T cells do not proliferate, indicating that the receptor can inhibit T cell proliferation. But if you add this one of the antibodies, you say the T cell can proliferate. So basically, this antibody can rescue T cell suppression mediated by this receptor. And this is the in vitro T cell activation assay. And you put this into an animal, and because there's no mouse homolog, so we took a mouse acute myeloid leukemia cell line, say 1498, that will overexpress the human B4 receptor, so the chimeric cell line. And then we inject it through the tail vein of the cell, and we treat this with antibody. And at day 27, we sacrifice the animal, looking for, so basically saying that this antibody, the control in black, the tumor is much bigger, and treated with antibody, you have reduced the tumor size. And this is demonstrates that this receptor is an immune checkpoint inhibitor receptor. By antibody treatment, we can rescue T cell activation. And the next thing is, where I told you, where this receptor is expressed cancer cells, they can sustain cancer cell growth and migration and infiltration, so function as oncogene. In this case, in vitro study, so if you, in vivo study, so if you, this human leukemia cell line, if you knock down the B4 receptor, and then you give the wild-type cell line, the knockdown cell line to an animal, you're looking for the migration of a cell called homing to different immune organs, bone marrow, liver, and spleen. You see the wild-type cell, the cancer cell can move to all these different organs, but if you knock out the receptor, this homing or migration or infiltration of these the cells are inhibited to show that antibody can, this receptor sustain that. And this is a knockout cell line. But now if you take the wild-type cell line, it's the same thing here, right? But if you treat it with the antibody can also inhibit the leukemia cell homing. So this show that this antibody functions by blocking the oncogenic activities of this receptor. So let's look at the efficacy model. So you put the MOL cell to the tailway, and in this case, we already treated the animal once. And you say with control at day 40, many of the cells begin to metastasize into the whole body at 37. But one dose treatment of this antibody, you can block the leukemia cell migration and tissue infiltration. And this can, of course, prolong survival. The antibody can prolong animal survival. And more importantly, this antibody has dose response. If you with this actually is 5 milligrams per kilogram, high dose, is a single dose study. So this study actually is a titration study. We say 0.01 milligram per kilogram, very low dose. You already see some efficacy at day 14. But at 1 milligram per kilogram, at day 42, you still see efficacy. So this is very good news, actually, to show that this could be potentially a drug candidate. And next thing we did was, I told you this antibody is an IgG1 molecule that can engage immune cells to mediate immune effector function. So we see if this if ADCC, antibody-dependent cellular cytotoxicity, play a role in the efficacy of this antibody. So what they did here is you mix the cancer cell that expresses the receptor, and then you mix with isolated human natural killer cells. That natural killer cells mediate ADCC, and sure enough, the antibody in vitro can mediate ADCC activity, and similarly can also mediate ADCP activity. ADCP stands for antibody-dependent cellular phagocytosis. Basically, the macrophage can engulf cancer cells. In this case, you mix with either human macrophage, mouse macrophage cell line, with a cancer cell, with antibody, you say, mediate about 20% of ADCC compared with the background, which is about 10%. So this is statistically significant. So to test these in vitro studies, to test this ADCC, ADCP immune effector function play a role in the efficacy of this in vivo. So we actually generated an antibody by mutating the asparagine 297 residue is antibody carcinogen. So this is the asparagine actually is glycosylation site. If you mutate this amino acid to alanine, the antibody cannot be glycosylated. So the non-glycosylated antibody cannot mediate immune effector function, cannot engage NK cells or macrophage. So we just demonstrated that you mutate this, it did not impact interaction with the receptor, which is result in the variable region of the antibody. And then you give this antibody to the animal, and we say if you knock down, if you mutate this asparagine residue, and basically knock down, eliminate the immune effector function, you basically reduce the efficacy. So the cancer cells can homing or migrate to different immune organs, like the bone marrow, liver, spleen, and PBMC. And put this. So next thing we did was this part of work was published last year in Cancer Immunology Research. If you're interested to look into the details. So the next thing we did was this is actually is a receptor, is expressed on cancer cells. Basically, say maybe this also can be constructed as a CAR T construct. So we did that. We made a CAR T vector, and we gave this construct to the animal. And you say that control PBMC, you say the cancer migration metastasis, and the control construct show the same thing. but the CAR T construct using one of the other antibodies showed remarkable efficacy. This was published last year in molecular therapy. Lastly, what we did was see this. We know that the antibody can immediate receptor internalization. If that's the case, we thought that this could be used as an antibody-drug conjugation because when antibody bind to receptor, the receptor antibody complex can internalize, can deliver the drug molecule inside the cell to kill the cancer cell. So we made antibody-drug conjugation. This actually is a work published in our lab in collaboration with KLG Tajukama. And we have a novel antibody-drug conjugation linker chemistry. And we basically conjugate 4 different molecules in one antibody. To make a long story short, we say that the ADC actually works better than the linked antibody. So due to time limits, I rush you to all these different. So just summarize this slide to show this is just one target, right? LRB4. And so we were able to take advantage of the multiple mechanism of action that contribute to the anti-acute myeloid leukemia efficacy in multiple animal models. So first one is that is a cancer cell expresses receptor that can inhibit T cell activation. So this antibody can rescue T cell activation. This is one mode of action. Next is this antibody can block this immune cancer cell migration and infiltration into tissue. So antibody can block that. So basically function as an inhibitor of oncogenic activity. And the antibody can, as an IgG1 molecule, can engage macrophage-mediated ADCP activity, phagocytosis. So this similarly is efficacy receptor engagement can also engage immune cells such as natural killer cells to mediate ADCC, that's antibody-dependent cellular cytotoxicity. And this start here, and this antibody, when receptor bound to the antibody, can mediate receptor internalization, so we can make antibody-drug conjugation ADC as a mode of action, and also as a receptor, we can also make a CAR T constructs. So demonstrating all these 6 different modalities or modes of action. So when we make a discovery, so we characterize the orphan receptor. You can publish a good paper or good papers. And the goal is if you can validate the target to make an antibody, can make a drug candidate, has potential for drug development. So so this is summarize this work in collaboration with Alec Zhang. And we made a significant discovery in different publications. But more importantly, we actually, while we're working on this, we actually launched this drug to a company in Palo Alto, California. So during the last 3 years, they already developed this drug, and clinical study will start later this year. So this is one example of how to translate very basic discovery to drug development. The second example is actually in collaboration with Anil showed at MD Anderson here in Houston. We identified a novel target, angiogenesis factor EGFL6, and this protein can mediate tumor angiogenesis, no impact on wound healing or normal tissue angiogenesis. And so this drug actually also launched to a company last year, and they already finished most of the preclinical animal tox studies. The clinical studies will start in 2021. And another example is working with Zhiqiang at University of Health Science Center San Antonio. Jing working on this channel, actually is a connection channel 640. These channels, they call hemichannels on one side. If 2 hemichannels dock to each other, they communicate between cells. And we generate antibodies only target these hemichannels, do not impact the whole channel. And we have one antibody keep the channel open all the time. And we have another antibody keep the channel closed all the time. Same target, but different antibodies. As the antibody keep the channel closed, actually works very well in spinal cord and other neuronal injury models. And the antibody keeps the channel open works in multiple animal models for breast cancer bone metastasis. So last year, 2 years ago, we got a $6 million grant from DOD to work on this breast cancer side. And also, at the same time, 2017, 2 years ago, we already launched these 2 antibodies to a company. And so this spinal cord model already started clinical trial in Australia last year. And the bone metastasis model will start in the US this year. And just lastly, I show you that working with Philip Sher, uses also actually his lab working on Modalities, we discovered a novel adipokai called eutrophin, and we published a series of papers. But more importantly, we developed antibodies by targeting this novel target for treatment of solid tumor, and more importantly for liver fibrosis. This is a late stage negotiation with a large deal. And last but not least, last October, we published a paper in Cell Metabolism, actually made the antibody by modulating laboratory level for treatment of obesity. And this actually also very hopeful that this will be a very promising, safe drug for treatment of obesity. So what I did here today is to walk you through very quickly the antibody drug discovery landscape. And so our lab can do this engineering studies, generate antibodies. But the critical thing is we collaborate with academic labs in Texas and in the country. And we actually access to the best biology, the best animal models, and discover novel biology, validate those with antibodies probe. And some of these antibodies became drug candidates. And I talk about biomarkers, clinical trials, and of course, due to lack of drug targets, so we need access to GPCRs, channels, transport. Actually, you talk about drug targets for small molecules. GPCR is most used for drug targets, but not for antibodies. So this is where Ross going to dive deep into this area, and we can access to more targets. And so in summary, I told you about drug targets always discovered in academic labs, also biomarkers. And the industry are good with development, they are not good with discovery. So the idea would be if the two can come together early and collaborate often, so we can make true innovative drugs. Actually, this seminar series is the innovative drug discovery development. So we want to, so there's our group called the Texas Therapeutic Institute, play a small role in bridging the two. And these are the people actually, Alec Zhao's lab, Nie's lab, and Kelji's lab, we work together. And me as a postdoc in Alec's lab, working on the biology of the B4 receptor. And Mark is a postdoc in my lab, mostly working on the antibody part. And Samuel working on the CAR-T, is a MD registered fellow. Heyu working on also the CAR-T. And Aki is a chemist working on ADC. And this is actually an old picture of our group 2 years ago. We are actually getting bigger now with all the funding. And thank you so much.